So, hello again. Actually, I'm happy that Nasser has accepted, because it is with this valorization bureau that I could make my success story possible, thanks to the support of INSERM transfer. So here I have something to declare as a conflict. Not before, but now I have something to declare, because I do participate to the work and business of a company which is called Tridec One. As we said this morning, in atherosclerosis and atherothrombosis, inflammation comes like a response to injury, and this injury can be either occurring in large arteries, and then in that case you mainly assist to dissection and eventually rupture, which may evolve to aneurysmal dilation. And in middle arteries, you are more often confronted with occlusion due to the thrombus that forms over a dissection or intimal flap. In both cases, you have an inflammation and an immune response that eventually might become chronic, and then it will be more and more sophisticated, with the adaptive immune system arriving right very close to the artery. You see, this is a normal aorta. In the normal aorta, you mainly see the media. You don't see the endothelium, and the adventitia is something with no boundaries. In a pathologic aorta, this is coming from an aneurysm that was repaired surgically, and the exceeding tissue was thrown away, so I was taking it from the trash. And in this type of diseased arterial wall, you see many layers. The media is here, and it is fairly destroyed. On the luminal side, you have at least one layer, which is the plaque, the atherosclerotic plaque, and you define it because of the cholesterol cleft inside. And surmounting over the plaque, you usually find some fibrin and what we call a mural thrombus, a thrombus that is growing on the wall, mural thrombus. In the adventitia, here you have this massive accumulation of leukocytes. So you see the inflammation is taking place mainly here, not there. And here what you see is that these inflammatory cells, these immune cells, aggregate, form some structures which look like well structured. And we can observe this type of formation either in large arteries that have undergone complication, like dissection, or in medium-sized arteries that mainly undergo occlusive complication. For instance, in myocardial infarction, coronary arteries, stroke, and peripheral arterial disease. In both cases, you see that there are immune cells, but the fate of the healing process and the fate of the disease is opposite. Here you have a dilative remodeling, and here you have rather a constrictive remodeling. So when I entered INSERM, my question was, is this immune response the same? And if it is so, why there is a different outcome? Or is it the immune response different, and which might explain why the outcome is different? So in order to apprehend this question, I started to study very systematically by immunohistochemistry and by FACS analysis what was the composition of these structures, So by immunohistochemistry, I tried to check whether, since they look like lymphoid organs, I checked for the presence of B cells. CD20 is enriched on B cells, and most of these cells, they are B lymphocytes, like in lymph nodes and in spleen. And these B lymphocytes core are surrounded by sparse CD3 T cells. Many of these B cells, they do produce immunoglobulins, meaning that these immune organs are functional. They do make immunoglobulins on site. And there are also some phagocytic cells, macrophages, dendritic cells, and other phagocytic cells which express CD68, which is enriched in lysosomes. And then I checked for the vascularization, because for an organ, a lymphoid organ, you need an entry with the blood, and you need an exit with the lymph. So I was looking for lymphatic and blood vessels. And for blood vessels, I used, as everybody does, CD31. So I could find CD31 lining the endothelium of the arteries, which is normal. There you have endothelial cells. And then I found a lot of spots which were not capillaries, which were not venules, were not vessels. They looked like cells. And even very, very close to these organs, within the lymphocytes, I thought I was looking at lymphocytes that were CD31 positive. So I thought it was an artifact, and I bought another clone, and then another clone, and then a polyclonal, and then many, many CD31 antibodies, and I always got the same results. Oh my God. So I started to do the literature. I should have started from that. And I discovered that in 1990, the year when CD31 was cloned by Newman on endothelial cells and platelets, that's why it's called PCAM, platelet endothelial cells, it was also cloned by Stockinger on leukocytes. But Stockinger was much less famous than Peter Newman, and this was completely neglected, forgotten. People don't know that CD31 is constitutively expressed by all leukocytes. But then, when you do a FACS analysis, you can see here is the granularity, side scatter, here is CD31 expression. You can see that innate immune cells are all positive for CD31, whereas lymphocytes, adaptive immune cells, are like split in two, a population which is positive, even though to a lesser extent than immune cells, innate immune cells, and the population that looks like negative, or at least much less positive. And so I was triggered now, because Stockinger says that all leukocytes express CD31. So how does it happen that in the periphery you have some cells that don't express it? Actually, these cells are memory T cells, because when T cells become activated and then memory, at each cycle of activation, this was shown by Demeur, they lose a little bit of expression of a CD31. It's like they were losing something that was preventing them to be activated. So, if I look at CD31 negative T cells, lymphocytes, in the peripheral blood of patients that have immune problems, in my case, the most important immune problems was atherosclerosis. What do I find? So I went to the bedside. I started with the bedside, looking for this population in the tissue, in the blood, as I was able to do already, and in the tissue. In the tissue, up to then, I was doing immunohistochemistry. But now, if you want to do multicolor analysis, multiparametric analysis, you are very easily, you run out of antibodies, because you can do 3 or 4 combination maximum. Whereas with the FACS analysis, you can go up to now even 40 with the CyTOF. So what I did was to go back to the trash of the surgery and take the aneurysmal walls, digest them with the enzymes, and do a single cell suspension, and check for the expression of CD4 and CD8 on one side, and CD31 on the other. Many other markers were there, but here I show you the T cells, helper and cytotoxics, and their expression of CD31 in the tissue and in the blood. So you can see that in the blood already, most of the cells that appear like CD31 negative, these cells, they are of the CD4 phenotype, and much less CD8 phenotype. And in the tissue, it is exclusively CD4. So T helper cells that infiltrate atherosclerotic, atherothrombotic tissue, they have lost their CD31. How does it correlate with the severity of the disease? Because in clinical research, mainly what we can do is association. Does the parameter A correlate with the parameter B and the outcome? So here what you see is the correlation with the size of the aneurysmal wall in patients that were operated in their blood. If the aneurysm was medium size, small aneurysm, big aneurysm. And you see that the number of CD4 T cells that had lost CD31 in their blood was increasingly higher in parallel with the severity of the disease. So this must be not a good thing to have a CD31 negative T cells in the blood. The same was found, even though I never published it, but the same we found in coronary artery disease. In patients with stable angina, acute coronary syndromes, and a patient that had chest pain but in the end had normal coronary arteries, it was the same trend. And the same trend we could find in mice, because if you leave knockout mice live long enough to develop a very complex lesion, even in the aortic sinus, you can find thrombus, small fissures and thrombus. And so the presence of a thrombus, which occurs in about 10% of the mice aged more than 1 year old, was correlated with a higher proportion of these CD31 lost T cells. So what does this CD31 do? And now I had to do some more serious bibliographic research because I needed to understand what does the molecule do, not just the marker, but what does the molecule do. And this was the beginning of my experience as a real basic scientist. So let's work from the beginning. PCAM1, as I said, was cloned in 1990 by Peter Newman and William Muller. And it was called PCAM because it was mainly found on platelets and endothelial cells. At that time, we knew several molecules that structurally resembled to CD31 in the sense that they are Ig-like glycoproteins, transmembrane glycoproteins. And since CD31 was found enriched at the junction in between endothelial cells, and it looks like an adhesion molecule, well, then it is yet another adhesion molecule. That was an assumption like this. 10 years later, almost 10 years later, in 1999, the first knockout mice were available, and it was evident that adhesion, leukocytes here in venules, is not an issue. Adhesion works very well in CD31 knockout mice, perhaps even better. If you look at the leukocytes here, they look like they are trapped close to the endothelium, much more than the other cells, the other mice. So adhesion is not invalidated in the absence of CD31. If this was an adhesion molecule, then it must be redundant. So the title of this paper was a redundant role for CD31. But actually, this is because we forgot completely that CD31 is also expressed constitutively by all leukocytes and platelets. So if all these people express CD31, and this is an adhesion molecule, everybody should stick to each other in the blood. We should have a jelly, not circulation. And this is likely not the case. And with the CD31 knockout mice, the first experimental works came up. And what we found was that any inflammatory model, or even thrombotic model, where is it? Thrombotic model, here, were exaggerated. In the absence of CD31, you have exaggerated inflammation. You have exaggerated aggregation of the platelets. And this is due to the fact that this is a receptor, and a particular receptor, because in its cytoplasmic tail, the tyrosines that can get phosphorylated, they are part of a motif which is called ITIM, immunotyrosine inhibitory motif. Why is it so? Because these motifs, they recruit phosphatases, not kinases. And phosphatases uncouple tyrosine kinases pathways. Mainly platelet and leukocyte activation pathways are kinase dependent. So to bring by phosphatases will uncouple them. That's why in the absence of CD31 you have exaggerated activation of leukocytes and of platelets. So CD31 is an Ig-like receptor, not adhesion molecule. And actually is a co-receptor because it is not able to signal by itself. It needs co-signals with other people. And it is a type of a receptor which is ITIM, which is quite specific type of receptors. The ITIM, generally speaking, uncouple ITAM signaling, because in this case the tyrosine is activator motif, and here is inhibitory motif. And so if you bring by phosphatases, they will take out the phosphor from the kinases, and the activation pathway is uncoupled. This is how they work. And CD31 is particular because it is transhomophilic. So platelets, endothelial cells, and leukocytes, they can engage their CD31 by just interacting in the blood, in the circulation. And that's how, by interacting in the circulation and engaging with CD31, each of them get mutually inhibited. And here I have tried to resume what CD31 does according to the literature before I arrived. And overall, we can say that kinase-dependent functions are uncoupled, whereas a phosphatase-dependent function, they are favored because CD31 breaks by phosphatases. And you have some signaling pathways which are dependent on the presence of phosphatases. And globally, overall, it happens that on endothelial cells, CD31 is rather a promoting coreceptor, a positive coreceptor, whereas on T cells and platelets and leukocytes, it's more a negative coreceptor. Actually, in 2002, Brown and collaborators in Birmingham, they had shown already that CD31 is responsible for mediating the detachment of leukocytes in between themselves. So in the circulation, what happens is that when the leukocytes meet to each other, they drive mutually an active detachment signal. And since this is an active signal, it is energy dependent. And in this paper, at that time, it was difficult to say that CD31 was not an adhesion molecule. So what they say was that when leukocytes are apoptotic, they cannot use anymore the detachment signal driven by CD31 because it is energy dependent. And this was the only way to get it published in Nature. Actually, this is not really true. The CD31 is not really missing on these cells. It is cleaved. Most of it is gone, but a portion which is very proximal to the membrane is still there. And I could see that by using two antibodies at the same time. Monoclonal antibodies directed against the domain number 1 and domain number 6. So Giulia Fornasa, who did one of my PhD students, she did this type of experiment, and she could find that, especially on memory cells, this population is enriched, meaning that they look like this. They have a CD31, which is only positive for this antibody and not for the other one. Therefore, this portion is missing. They are truncated. And then we went back to the literature again. We have not learned everything all the time. So we went back to the literature and we could find that this was already described as well. The shedding of CD31 on activated endothelial cells, leukocytes, platelets was already described before us, but nobody paid attention to that. The shedding occurs with activation. Why so? Because when the stimulus is strong enough, you can bring all the phosphatases that you want, but eventually the activating receptor will get through. And if the cell activates, the proteases that are also activated at the time of activation, they cleave a lot of membrane proteins, including CD31, and not exclusively CD31. For instance, you can measure a soluble P-selectin as an activation marker of endothelial cells and of platelets. This is made true because proteases cut it at the surface of activated cells. And the same protease can cut many of them because they just act in a non-specific way behind the lysine or arginine. So what we do now, I wear back my hat of medical doctor. So this is a nice story, but what do I do with it for patient? Can I translate this to clinic, to have a clinical application? Well, we tried, and INSERM Transfert was very supported, to patent the use of the detection of this portion as a biomarker and to exploit the presence of a truncated portion as a therapeutic target. Because this portion, the proximal to the membrane, I just go back a little bit. Too much animation. This portion here you see is the most important portion for the signaling, because the receptor must co-cluster with items. And to do so, there are some homotypic sequences very close to the membrane that allow the sticking of the molecules in between themselves. So the fact that this portion was still there gave us an idea. Perhaps we can still signal because this portion is still there. And actually, when you have the activation of lymphocytes or another leukocytes with another cell, what happens is that the CD31 that is distributed all over the cell in a resting state, under activation condition, here we created an immunological synapse by adding a super antigen, and the antigen presenting cells and the T cells, they form an immunological synapse in this condition. In this condition, what we observe is that CD31 is clustered at the site where the TCR, the immunological synapse, is formed. But if you look at the two cells on the side, you cannot see that indeed within the clusters of the TCR, within the synaptic portion, the most distal portion of CD31 is gone. There are holes. So you have an intact molecule all over, and it is like this is necessary to limit the spread of the activation signal. Instead, in between the synapse, where the cell must do the exchange with the other cell, the CD31 is cleaved. And I can say that it is cleaved because with the other antibody, the red one, you see that the holes are filled up. And it was shown 30 years ago almost, that an antibody, the LIP21, which is directed to a very proximal portion of the membrane, the one that I still find, the one that is spared by the cleavage, an antibody that recognizes this site of CD31 exerts a very strange effect on T cells. Here you see T cells that are activated in a round bottom well. They get clustered at the middle, at the bottom. You can see they are activated even without counting them. But in the presence of this antibody directed to this portion, you see that the cells even face against the gravity. They are not gathered in the middle of the well. It's a round bottom well. They are spread all over, like they were trying to escape to each other. Actually, what happens is that the mutual inhibition of the cells is engaged by the antibodies. The antibodies directed against this portion are engaging the receptor in a permanent way, so that even if they were activated, they still receive the detachment signal, and they detach from each other, because the cells are not dead. They have just cleaved their CD31, so they have energy to do the function. What is interesting, and that's why I decided to do some translational application, is that the epitope of the antibody did exactly the same. The authors used the epitope of the antibody in order, I think it was a control, in order to check whether the antibody was specific or not. And they have also used the peptide alone in cultures. And they saw that the proliferation, count per minute with the tritiated thymidine, the proliferation of T cells went down in a dose-dependent manner, not only with the antibody, but also with the peptide, the epitope of the antibody. They couldn't understand how it worked because the cells were activated and they looked CD31 negative. But at that time, they published all the data in an honest way, and I regret that time because if they didn't publish the results they couldn't explain, I couldn't know. So what we did was to use this peptide, which is also active in vivo. And in vivo, what it does is to spare the life of mice that have been grafted with the bone marrow. With this type of immunointervention, what you get is the death because of graft versus host disease. And the mice that received the peptide were spared. Their life was spared. So it's a big immunosuppressant. And the sequence of the peptide used by Zender was almost completely homologous to the sequence of the mice. And the most conserved portion was really close to the membrane. This is the membrane. So what we did with Julia again was to use this peptide in atherosclerotic model, and we could see that the peptide, here it's labeled in red, was targeting cells within the atherosclerotic plaque. What did I do? Oh, okay. Within atherosclerotic plaque, and these cells were mainly T cells and macrophages. And what we found in the blood and the tissue was that there was a mirror change in the ratio between effector T cells, T cell activated, expressing CD69, CD69, CD3, CD69, and Tregs. where with the peptide, the white bar columns, with the peptide, the mice had less activated T cells and more Tregs. And this could be possible because the Treg pathway is phosphatase dependent. The GAB-ERK-MAP kinase pathway that makes FOXP3 expression possible is phosphatase dependent. Importantly, since we wanted to make a drug now, we should be sure that the peptide does not affect resting cells. So here the peptide is green, and you see that it can only stick to T cells upon a TCR cross-linking, which is a way to activate T cells. Whereas resting T cells do not bind the peptide, And in functional tests, when we checked for the phosphorylation of the item of CD31, we could see that the presence of the peptide could uphold the phosphorylation of the item, so it was an agonist of the receptor, only in the presence of a stimulus, the TCR cross-linking. Whereas if we put the peptide alone, it doesn't do nothing. It doesn't bind, it doesn't do nothing. That cannot, actually the peptide cannot do nothing because the CD31 can only get phosphorylated if another stimulus is occurring, if activation is ongoing, because it is not able to phosphorylate itself. It needs another kinase to do the job. So now we went from the bench. I was convinced that I could make a drug. The dream of a medical doctor that does basic science is to make it concrete one day. So I went from the bed to the bench, and now I want to go to the pharma. Insem transferred, can you pay? They paid a little bit. They supported this type of experiment that I did in my small garage, because this type of experience must be done in a large scale. But I tried to do it at a small scale. What I did was to construct a library of different peptides in order to check for the best hit, for the best lead, the minimal sequence that was active and that could be druggable. And I also had to do in a way that the patent from Zender, the one that discovered the peptide, could be circumvented. Otherwise, I couldn't make my own patent and my own drug. So I had to screen for the peptide and look for something that could be patentable and druggable. So to screen, I took the blood of healthy donors, my coworkers, and I checked by fax analysis, an automated fax analysis with the plate reader, for surface marker of activation and soluble in the supernatant markers of activation. This was very effective because with 1 tube of blood, I took the Buffy-Cot for T-cell analysis and the platelet-rich plasma for platelet analysis. And the reading was surface CD62P and 62P and CD69, and I read almost the same molecules in the supernatant. So then, in order to choose the best hits, what I did was a hierarchical clusterization. I don't know if from a statistical point of view that is solid, but what I tried to do was to check for those families of peptide that were good for all the markers, for the 4 markers, that were good to blunt platelet activation and T-cell activation detected in 2 ways. And so there was a family of a peptide which led me to patent. The first patent was called PepReg, regulatory peptide. But PepReg was not perfect because it's insoluble. Barely half of it is soluble and the rest is aggregated. So we had to find something better. We did another patent, which I called improved peptide. And this improved peptide, actually, I just cut one arginine here and one lysine there, because by cutting this arginine here, I rendered it more soluble, and by cutting this lysine here, I rendered it less anaphylactogenic, because the cationic peptides are anaphylactogenic, and then for a drug, it's a problem. And then I did a lot of experiments with this lead, but they were not reproducible. And the people that made the peptide for me, they said this is because they are L amino acids. Try to use D amino acids. D amino acids are used by bacteria, not by eukaryotic cells. So we don't have eukaryotic people, individuals, don't have the peptidases to cut them. So what was suggested to me was to do a retro-inverso peptide. Retro means that N-terminal and C-terminal are inversed. And inverso means that the isomers from L becomes a D. By doing a double mirror you almost get the same molecule because it's mirrored twice. When you flip vertically, flip horizontally, you have almost the same figure. And the P8-RI peptide of 8 amino acid retro inverso is highly soluble in water. And it was at least as much effective as the other one. So we went on with another student, and we went on for a therapeutical use in an atherothrombotic complication. It's a model of dissection, aortic dissection, the angiotensin II apoE knockout mouse model. In this model, you get a dissection and then eventually a dilation and an aneurysmal transformation. And this aneurysmal transformation was limited by the use of my peptide. Why so? Because with the peptide, I favored the differentiation of macrophages towards arginase 1 positive cells, M2, whereas I blunted the differentiation of macrophages towards the M1 pro-inflammatory phenotype. By doing so, in vivo, within the lesions, these are cross-sections of the aorta, and the half moon that you see here is the false lumen. Inside this false lumen, you could see that there were leukocytes, innate myeloid leukocytes, CD11B, and these leukocytes were arginase 1 positive or not, iNOS positive or not. So I could see that in control mice, mainly the leukocytes were expressing iNOS and very little arginase 1. So they were mainly M1 pro-inflammatory macrophages. These macrophages, the pro-inflammatory one, they degrade the tissue, they don't build up. Whereas with my peptide, there was more arginase 1 and virtually no iNOS, meaning that the macrophages were forced to switch to M2 phenotype. And this can be explained because the switch is regulated by phosphatases. And by doing so, with the peptide and with the macrophage of the type M2, we could accelerate the healing of the aorta. And therefore, we could prevent the dilation, the aneurysmal transformation. And this was the first publication after the patent. The patent was published before. And Francesco could exploit everything. So, we have a novel type of drug, which is not an immunosuppressor. It is an immunoregulator, and which is not targeting one cell, one pathway. It is targeting many cells, many pathways, but in a kind way. It's not it doesn't block anything. But what it does, CD31 engagement, is to raise back the threshold for the activation, because it maintains the phosphatases into the lipidic raft. I can go fast here. To be able to know if I was right, I used a model where different type of cells are involved into the immune response. This is the model of delayed type immune response, hypersensitivity. With this model, what you do is to prime with an antigen on one side, for instance, the skin of the abdomen, and 5 days later you challenge on another side. So if there is a response, it means that there is an adaptive immune response, which is able, it has been mounted in lymphoid organs, and is able to act anywhere there are memory cells. So with this challenge, what you do is to measure the ear thickness, because if inflammation is taking place, the edema will swallow the ear. So it's very convenient to measure. If it is thick, there is edema, there is inflammation. If it is not thick, there is no inflammation. So what we did was to use different doses of the peptide, or no peptide at all, or a scrambled peptide, or we used the peptide in mice that were not primed. What you see here is the second reading as compared to the first reading. In the absence of the peptide, the ear was swallowed. Whereas with the peptide in a dose-dependent manner, you had less and less thickening. And this is, therefore, one proof, indirect proof, that with my agonist I could modulate the dialogue in between the different cells that are all involved into the DTH model. So if we can resume, we can say that the immune system is made in a way that the cells must talk to each other. You know, you need an antigen-presenting cells, a T cells, and then another cells, the B cells, to make the antibodies. And you need danger signals that have to act in the same time as the trigger. And everything is made in order to be sure that the immune system acts against the pathogens, not against yourself. Otherwise, you develop autoimmunity. And CD31 makes this important task to moderate, to orchestrate, to be the chairman of the dialogue in between the different cells. When there is an immune response that is not appropriate, like in rheumatoid arthritis or multiple sclerosis and atherothrombosis, we found that there was a pathologic cleavage of CD31 which was responsible for an hyperactivation of the different type of cells. The cleavage of CD31 is associated with an inappropriate activation of the leukocytes in the long run. When there is the stimulus, it's normal, but if the cleavage is persisting afterwards, this is not normal. The difference with the other immunosuppressors is that if you use steroids or other immunosuppressors, you completely erase the immune system, and then the pathogens can win. The secondary effects of immunoblockade are heavy. Most patients now, they suffer more of the infections and cancer because of the immunosuppression in the chronic inflammatory disease. So this was a long story to say that it's possible to go from clinical studies and practice to basic science with enthusiasm, and then with a similar enthusiasm also to make a pharma biotech. The pathway is long, but it is an enthusiastic one. So now I created my startup. Welcome to TriDec 1, the future of immunomodulation. Then you have to sell yourself a little bit. And why TriDec 1? TriDec, it means 30, so 31, 30 in Esperanto. And here I took this from internet because I guess that young people don't even know what is Esperanto. Esperanto was a language that was taught by a Polish ophthalmologist. It was a language meant to be so easy that people from anywhere can talk to each other, like platelets with leukocytes and endothelial cells. And so since CD31 can make these people talk all together, I thought that the company could be called Esperanto 31. And you see here there is a 31. Nice. So the nice part of this adventure is that I think that you can do nice science, nice translational science, and also be happy in doing a pharmacological drug company. I don't know you, Peter, but myself, I don't run the company. I'm not able to do that, but I'm happy to participate in that. And the CD31 agonists, in our case, what they do is to moderate the dialogue in between the different immune cells to keep everyone calm. Thank you very much.
Thank you. Thank you very much. So we are in good time. So any questions, please? While you are thinking, maybe you could tell how the enzyme tech transfer office helped you, because that could be a good opportunity. Also, if in your university there is no tech transfer office, actually, who has tech transfer office? Or who knows that your university has tech transfer office? Can you raise your hands really? This is a little sad, yes. So
I think a lot depends on the mentor. Because the previous mentor I had, they never talked about valorization and patents. And then I happened to be in a laboratory where this was a routine. My colleagues were talking all the time, we can make a patent, we can make a patent. And I said, what is a patent? What do you do with it? And then I started to get interested into that. And when I asked for help in cell transfer, I think it's the same in the foreign laboratories as well, in the valorization bureau. Actually, there are many scientists. There are scientists doing this job of bridging the gap in between science and the technology transfer or product transfer. And so I was really helped by these scientists, because in order to write correctly the patent, they need to understand correctly the science and we were discussing science most of the time. But then I learned from them how to foresee the possible hurdles. In the experiments I could do to try to include all the controls that could be required, and also in the way of vulgarizing the results. Talking about CD31 agonist, not the sequence of the peptide, for instance. And then you can go to the conferences, no problem, as long as you don't disclose the secret that you must protect.
Yeah. So, questions?
Thank you very much, Pina. Exciting. Is it possible to anticipate the duration of the treatment to be active in one specific disease? For example, the mode of administration and possibly the price of 1 week treatment, for example.
Yeah, it is possible. And this is the reason for which we went for peptides and not antibodies. Because when you go for antibodies, you may expect that the effect will last long. And we might not want that, because if you get an infection, if you know that you have a cancer, then you don't want to wait 3 months in order to be again immunocompetent. So with the peptides, what you have is the half-life of the peptide itself, which is not very long, not short either, it's 4 hours. And so you have to give it every day. And we target immune disease where there are waxing and waning phases. And our target is the waxing phases, when the patient is really suffering a lot. And we try to do steroid sparing effect, so that you can diminish a little bit the immunosuppression and help with the CD31 agonist. So you can target short window time with the peptide, and you can hope that in case of need, if, for instance, you need to combat tuberculosis or another bacteria, the type of immunoregulation is mild enough to allow the system to work in case of need. So that's why we went for peptides. It's a short time window. But then you can imagine also to work on the structure of the peptide. We talked about bicyclic peptide with AstraZeneca before. You can make it resistant so that it cannot be eliminated by the kidney so quickly, if you wish to have a longer lasting effect. So if the pharmaceutical company grows up, we can have a family of different type of drugs with longer or shorter half-life. And then it can be a depot. It can be a long-lasting also administration or not.
Okay, thank you very much. Yes, a question back there. Actually, you can come up to the microphone anyways, but yeah, I'm happy to bring it.
I take note of the information for the next one. I'm coming from the field of the intensive care and not cardiology. According to what you said here, it seems that you find a perfect solution to sepsis, which is an exaggerated reaction from the immune system to an infection. And you mentioned one of the work published in 2005 from Mars in co-workers.
There was a work in shock, yeah.
Exactly, showing that indeed sepsis was increasing.
It's aggravated. In CD31 knockout mice, they die, all
so
of them.
Could the story continue also in this direction?
Yes, yes, because then you have another effect which is very important. I didn't deal with it now, but it's mechanosensitive properties of CD31. And in sepsis, the thing that kills you is the blood pressure drop, is the vasoplegia. And then with the CD31 agonist, you can bring back also the regulation of those receptors in the vascular wall that are responsible for the adaptive tonus. So you have the cytokine storm under control, the intravascular disseminated coagulation under control, and the vascular tone back. It's the ideal drug, but we still need to make solid proof of concept for that.
Okay, thank you very much. I think that the take-home message for this session is that check out the website of your own university if you have a tech transfer office, and ask your boss that if he or she knows about it, and talk to the tech transfer people, okay? And also to your boss that be aware of the protection of intellectual property, and also that science has to be turned to business sooner or later, because otherwise we are just spending the taxpayers' money. Thank you very much.